So this is going to be a really fun discussion because we're joined by someone who's been on our program before, right at the start of the pandemic. Actually, was the last time we had him on the show. We have with us the chair of translational medicine for the University of Salford, Professor Michael Lisanti, who's going to be talking about his new paper that was just published in Nature, in the journal Nature, talking about how cancer power failure stops the spread of tumor cells. That's the line from the press release about this paper and what it means. Joining me on this panel to talk with Dr. Lisanti are my friends Tucker Goodrich, who we have on frequently on the radio program to talk about nutritional connections to mitochondrial problems and the metabolic dysfunctions that manifest from that. And then, of course, we have with us our physicists, in-house physicists for everything we go to for learning about the physical side of things on our program, Dr. Yu, who joins us as well. So thank you all for joining us today.
It's a pleasure as always.
So
delighted to be here.
Yeah, Dr. Lisanti, you know, a lot of things have happened since we last spoke with you, and we continue to follow your work with great interest. And I want to ask you just a little bit off topic for a second. You know, I've seen that you're one of the most cited scholars on Google Scholar. How'd you get so many citations? What drove all those different references and so forth?
I guess, you know, we're always interested in the unknown. You know, some people are interested in, you know, incremental discoveries, and there's nothing wrong with that. But the thing that drives me is the unknown. So I, just by luck, I guess, I always seem to find areas that are, you know, at the borders between two areas. So for example, cancer and aging, you know, there's the place where that meets is very unique, or autophagy and cancer. You know, there are all these areas of overlap between two important areas of science that people haven't investigated. And usually there's gold there in those places where, you know, there's a scarcity of information. I don't do it on purpose. I just like working on the unknown, you know, what is, and more often than not, I find that, you know, and the same thing is true for us. I mean, we make a lot of assumptions in our research about things that are known, but then it turns out that they're either really not known or they're wrong. So, you know, it's important to reinvestigate certain things because we have new tools, and, you know, science is always getting better. So we always have more sensitive methods or new approaches. And that's the case also with this story. So basically we were thinking that ATP was very important.
So before you jump right into it, I have a little question. You're involved with a company called LUNELLA Biotech. LUNELLA is, if I'm correct, Italian for moon girl.
It's Italian for moon.
For moon.
And they subsidize our research.
And who was that company named for? It seems like an odd name for a biotech company.
The truth of the matter is it's named after my daughter because her middle name is Luna. So it's Camilla Luna Lisanti.
Ah, and how did she come into this story?
I don't know. I can tell you, but you should Google her name, Camilla Luna Lisanti, or just leave out the Luna, but I don't think it's in the news articles, but you'll see if you Google Camilla Lisanti cancer antibiotic, this actually was all her idea. So what happened is that Federica, my wife, and I are both scientists, and we work together. So Camilla had been hearing about cancer for many years, and
around the dinner table, no doubt.
Exactly. So we would talk about homework and sports and gymnastics, and we'd always wind up talking about experiments. And, you know, Camilla looked bored. It was just fortuitous that we then tried to engage her in the conversation, and she'd heard it all before, so she didn't want to really talk about it. So she wanted to give us the answer, so we wouldn't have to talk about cancer anymore. And the answer was, we asked her how she would cure cancer, and she said it was obvious. This is when she was 7 or 8, that she would just use an antibiotic like when she had a sore throat.
From the mouths of babes.
Yeah, we began to think about that, and she was usually right, and she still is usually right. So we follow her advice, and so we took the idea into the lab, and it actually worked, because certain antibiotics have a side effect, and that side effect is they inhibit the production of new mitochondria. So it's actually a very safe way. When you think about, it's a very safe way to inhibit mitochondria. And one of the best examples is doxycycline, because you take it for 6 months at a time for acne with minimal side effects. And so that turned out to be very important, because that turned into a clinical trial in using doxycycline in breast cancer patients in Italy that was successful. It turned into our research program at the University of Salford, and it also spawned a company that funds our research to look at potential ways of inhibiting both aging and cancer. So I think Camilla deserves a lot of credit for that. And actually, at that time, we didn't know what to do. So we contacted, I had been talking with the Wellcome Trust about writing a grant. And in the end, I didn't get the grant, but I still, and I guess that was a blessing in disguise, because then, all of this happened. So, but I asked them, what should we do? It was Camilla's idea. And they said, you absolutely have to put her on the paper, because it was her idea. So we had to contact her school, her grade school, her prep school. And we needed permission, because she needed an affiliation on the paper. We couldn't put her, she had to be where she is. So
you couldn't list her as a representative of the grade school without their approval, of course.
Exactly. So they were delighted to have her, as a co-author on the paper.
So does that make her the youngest co-author on a published research paper? I would have to think she's pretty darn close. I don't know,
but she was, I guess, 8 or 9 at the time. She had invitations also to go on the BBC News, to go on, I guess it was the BBC One show. And it was actually snowing at that time in England, so she preferred to play in the snow. She turned down the BBC One show.
Invitation.
Smart girl.
So we still very avidly seek her advice about things, and so this has been a real whirlwind in terms of, we were always interested in mitochondria and cancer, and then we figured out that it was the cancer stem cells, so more related to recurrence and metastasis, and now we think it's
which is the current paper. But just for the audience, just to give them a little bit of background, if you're looking at mitochondria in the cell and then you start looking at antibiotics, a mitochondria is effectively a captive bacteria. So it makes sense that a bacteria would be, that what would affect a bacteria would also affect the mitochondria, which is of course what you see with doxycycline, which is, in addition to a treatment for cancer here in the United States, it's the common first-line treatment for Lyme disease. So
exactly. So mitochondria evolved from bacteria that were engulfed about 1.5 billion years ago. So so all antibiotics that affect bacteria may have mitochondrial side effects, but usually they're manageable side effects. Right. And what we're doing in this case, also with this paper, is we're taking advantage of the fact that these side effects are manageable and can then be used and repurposed instead of the side effect as the primary therapeutic effect.
So now you're specifically in your current paper looking at another antibiotic, one that's typically used to treat tuberculosis, if I'm correct.
Exactly. So there's something called the ATP synthase, which is if you look at the crystal structure, the 3D structure, you know, it's a nanoscale rotary engine. And so what
Yes.
it does is it actually rotates. So it takes the torque, the physical energy from torque, and converts that into chemical energy, which is the ATP. So it actually runs with an efficiency close to 100%. So it's actually the most efficient engine ever produced. And so that drug, the bedaquiline, which is also known as Serturo, that's the commercial name for it from J&J. It's an inhibitor of the bacterial ATP synthase, which is the one in tuberculosis. So it's used very specifically for drug-resistant TB.
Is this a macrolide antibiotic?
No, it's actually a different class. So it was the first in its class, and it was originally discovered by J&J. It's the only antibiotic in its class. It's an ATP synthase inhibitor, and they designed it specifically for overcoming drug resistance. So, you know, that's also a clue in and of itself that, you know, basically you can overcome drug resistance with an ATP synthase inhibitor. That's
Which is, of course, a huge problem in cancer treatment, is cancer cells become, or some cancer cells are resistant to the drugs you're using to try and treat the cancer.
Exactly. And the benefit of this drug, which we find actually intriguing, is the specificity. So we've tested other cell lines that are normal. For example, fibroblasts and also normal mammary epithelia. And the bedaquiline has no effect on them. But on the metastatic cancer cell line, MB231, it's a kind of mouthful, and also MCF7 cells, which are both breast cancer cell lines, it selectively inhibits ATP production. So it appears that it can actually sense what cell is a cancer cell, what cell is a normal cell, and then avoid the toxicity in normal cells and only target the cancer cells. So in fact, in fibroblasts, when we first published early findings on the drug, I think it was in 2016 or 2017, we saw it actually increased ATP production in normal fibroblasts. So that's just the opposite of what it does in the cancer cells. So that's remarkable. And that suggests that the ATP synthase may have more than one conformation. And so the drug may only bind to a particular conformation state of the ATP synthase, which may be different in cancer cells. And that's to our advantage, because then that would minimize toxicity.
So in these metastatic cancer cells specifically, because if I remember correctly, you found it had little effect on non-metastatic tumor cells, something in them has changed in the ATP generation that this drug specifically inhibits, whereas it doesn't do that on other cells, including normal cells.
Yeah, we came at it in several different directions at the same time. The first direction is we thought that ATP would be important. And so we separated the cancer cell population based on ATP levels. And for that, we used a dye. It's an ATP binding dye that's only fluorescent when it binds to ATP. So otherwise, it's not colored. And then we use something called a flow cytometer, and it's also known as a fax machine. So what it basically does is it allows you to sort cells based on their fluorescent parameters. So it knows, you tell it you want only the cells that are this color, and it'll pick those up and put them in a basket for you. So you can actually separate the cells using this dye and the fax machine into ATP high, ATP low cells. So then you can compare their properties. And this addresses an important issue in cancer biology, which is metabolic heterogeneity. So environmental stimuli, and maybe it's all related to post-translational modifications, or how the cells handle their metabolism. So because this is from the cell line, the same cell line. So we're taking the same cell line and stratifying it. And so we see that the ATP high cells have all the most aggressive properties. They're drug resistant, they're metastatic, they're more invasive, they migrate more, they have more stem cell characteristics, they have more anchorage independent growth. So that was one angle. Another angle was we wanted to know what was the protein that was key. And it seems to be a subunit of the ATP synthase, the enzyme that makes the ATP, the motor, the mitochondrial motor for ATP reduction. And that subunit is actually the one that does the torque. So the structure of the ATP is like a dumbbell. So you have one part of the dumbbell sits in the membrane, then you have that sort of a stick that goes in the middle, and then there's a second dumbbell. The second dumbbell makes the ATP, the second part of the dumbbell. And the stick that holds the two together is that gene that we figured out is most important because it actually provides the torque. So it's the part that spins. And without that, you can't put the motor together because you need to have that stick between the two weights on either side.
So it's like a wheel without an axle at that point.
Exactly. So we take out the axle and then you'll have no ATP. So that's
because it can't spin and go through the process of generating it. The old Krebs cycle that we all had drilled into us in high school biology.
Yeah, it's a very simple, actually, physical problem. So we're taking out the torque. And it turns out that this engine needs to be properly folded. And so where the bedaquiline or sortero binds induces the degradation of that subunit. It's called the gamma subunit of the ATP synthase. And so it gets destroyed. So then you can't put the engine together. You can't put the other pieces of the engine without that part that spins. So it's like, throwing a monkey wrench into the mitochondrial ATP production, but just in the cancer cells.
Putting a stick through the wheel of a bicycle. The wheel can't turn, bicycle can't proceed, right? So now, just to, again, go back a little bit for the listeners, isn't the basic idea of cancer that there's some genetic mutation that triggers cancer? It doesn't sound like you're working through that pathway at all.
Well, I think part of it is that you can have oncogenes and they can induce metabolic changes, but not always in the same context. So, for example, the original work of Bob Weinberg, they put RAS in cells trying to transform them, and actually all the cells died. So because RAS is an oncogene, it induces oncogenic stress and then oncogene-induced senescence. So the cell has to be immortalized first before it can become transformed. So it depends on context. So I think there is, and we don't know the answer to this, but there must be something different in the ATP high cells and the ATP low cells. We don't know what the changes are. Maybe they're genetic, maybe they're epigenetic, but we do know that a subpopulation is more aggressive, and that more aggressive subpopulation makes more ATP. And so that allows them to be the fittest cancer cells. So they're the energetically fittest. And we see that as a consequence, they have all these aggressive behaviors. And then if we pull the plug on the ATP, then they don't metastasize. So there are no FDA-approved drugs for metastasis, for preventing metastasis. So
which is, of course, the biggest problem in cancer is when it goes throughout your body.
Exactly. So 90% of all cancer patients or more, they die, 90% or more of patients that have failed treatment die of tumor recurrence and metastasis. So that's why the cancer can become such a lethal disease once it spreads. So the idea would be to prevent the spread, and that could potentially turn cancer into a non-lethal disease. But we need a drug that's not toxic, that could be then taken by the cancer patient to prevent the metastatic event. So once the horse is out of the stall, we don't know what happens then, but if we can prevent the horse from bolting, then we can prevent the metastatic event. And that would be great, because that would save a lot of human lives.
So
Right.
in your past research, you've explored in different ways how these doxycycline and azithromycin and some other things are effective at targeting cancer stem cells. What is it about this new Nature paper that you've published that kind of is, so, what is the next step that this has demonstrated that is different from the work that you've done before in terms of the big picture here?
Right. So the other drugs like azithromycin and doxycycline are mitochondrial biogenesis inhibitors. They inhibit the proteins that are important for the function of the mitochondria, ultimately generating ATP. So it's actually the same mechanism ultimately. But people could have said, it could be something else. It could be the act of mitochondrial biogenesis that is the most important, not the energy production, because mitochondria do lots of things besides make ATP. But that is one of their primary functions. In fact, if you look it up in the literature, we all make our body weight in ATP every day. So if you have the average 70 kilogram individual makes 70 kilograms of ATP a day. The cost of buying 70 kilograms of ATP is several $1 million. So every day, everybody's body makes millions of dollars worth of ATP.
So everybody listening to this should feel like they're worth more right now than they were a few minutes ago.
Exactly. And also ATP goes down with aging. So we think that loss of ATP is part of the root problem in aging. So the cancer cells have found a way essentially to escape aging by boosting their mitochondrial biogenesis. So we're just trying to normalize that. And in fact, these drugs, like the doxycycline and the bedaquiline, they don't affect the tumor growth, they only affect the metastasis. So what we're doing is really just turning down the volume on the mitochondria. We don't want to completely destroy the mitochondria, we just want to control the volume. And that, turning down the volume control may be sufficient just to inhibit the metastasis.
So you're obviously, these hyperaggressive metastatic cells require more energy. You're turning off their power supply.
Right. It's a very simple idea. We're just flipping the switch, the off switch. And the off switch involves reducing the mitochondrial production. And I think they're, for their survival, they're addicted to energy production. They need to make more ATP because they have such an aggressive agenda. So if they then do those terrible things like metastasis, they'll die. So they won't have the energy to do that. So we're just trying to turn down the volume on the aggressive behaviors and reverse, the drug resistance, inhibit the stemness, prevent the metastasis.
Right. You looked at, if I remember correctly, you looked at doxycycline in this paper, in your latest paper as well, and found that it wasn't particularly effective in this application. I presume where you just would have used that.
It's all relative, I think. So they were more resistant to doxy. They were still sensitive to it, but the ATP low cells were the most sensitive. So it's a dose effect. But the bedaquiline, I think, was especially good because it's targeting the root cause, which is the ATP. And
right.
Does that indicate that that's the root cause for a lot of cancers? Is this ATP pathway here that's been?
I think other people will need to repeat our experiments and expand on what we've shown, but we did it with 4 different cancer cell lines that were all independently generated from patients many years ago by various investigators. So it seems to be a general principle, but other people will need to reproduce the work. And I hope that the method we've developed will actually be something that, will be something that's passed on to other people, that, they will investigate this because it's a simple method, and the root, the ATP high cancer cells are like the needle in the haystack. So most drug companies will screen with the cell line as it is, not looking at the ATP high cells. But now with the ATP high cells, you could then directly address the problem of drug resistance, because you can isolate them before, so there's a whole argument in the literature about, it's a chicken and egg, cart and horse argument. What comes first? The resistant cancer cells, do they pre-exist before you give the therapy? Or does the therapy induce the drug resistance? What our data does is it resolves that chicken and egg question, because it directly shows that those resistant cells actually exist before the drug comes along. And what happens is that you're just expanding essentially the ATP high population, because they're the only ones that are surviving. So in many cases, the drug resistance is just a reflection of the fact that the ATP high cells are drug resistant.
Right, right. And the drug itself isn't doing anything to them. They're already that way. So one of the things that we hear a lot about in theories of cancer treatment nowadays is the Warburg effect, which sounds to some extent like it's related to what you're discussing here. Could you explain a little bit about the Warburg effect and how your treatment is either similar or different from that?
Yeah, we actually were the first to propose something called the reverse Warburg effect. And the reverse Warburg effect is the opposite of the Warburg effect. So what we proposed is that there are two compartments in tumors, at least. One is the stroma that actually is glycolytic, and the byproducts of that glycolysis, which are lactate, ultimately also ketone bodies, glutamine from autophagy. So basically the stroma, the connective tissue, is the Warburg cells, and they're making food to feed the cancer. And the cancer cells are actually oxidative. So they're like a parasite that is sucking all the nutrients out of the stroma. So it's the Warburg effect in the stroma, but it's actually oxidative metabolism in the tumor cells. And we studied that for many years, and that's all been validated now. And many people have seen it and published it again and again in Nature. And we actually wrote a review on that in 2017, where we highlighted this two-compartment metabolism.
So now just for the audience, the Warburg effect is, as I understand it, where the cells cease relying or rely less on mitochondria for power, and they're switching to an older power source, which is breaking down glucose through fermentation, which is why when you get a PET scan, they're looking at basically the cells are burning glucose through this fermentative metabolism rather than using their mitochondria. Is that correct?
Right. Basically, the Warburg effect suggests that cancer cells don't use mitochondria.
Right.
So, but, obviously there are cancer cells that don't use mitochondria, but the moral of the story is really that those are not the dangerous cells. So the dangerous cells are the ones that do use mitochondria. So the problem is that, I guess we've all been digging in the wrong hole for many years. And now we've found gold in another place, and that gold is in the mitochondria, because basically the most dangerous cancer cells have fully functional mitochondria. They're making the most ATP they possibly can, and they're the most dangerous cells. So we found the needle in the haystack that is the cause, or we suppose is the cause for the metastasis.
Or at least the energy source for the metastasis, which is almost as good.
Right, and it may also be that, those other cells, like the stroma, the non-dangerous cancer cells or less dangerous cancer cells, they may be making the food to fuel the metastasis. So I think everybody agrees that tumors are heterogeneous. So it's not just one type of cancer cell. There are many different types of cancer cells, sometimes 2 or 3 or up to 9 clones.
In a single tumor.
In a single tumor. And the tumor is very heterogeneous with hundreds of thousands of million mutations. And so it makes it very difficult in the end to know what is the driver mutation. What we're saying is you don't need to know, you can use a kind of black box approach. You don't need to know what the mutation is. You just need to know, what are the ATP high cancer cells and how to eradicate them. And so you can take it it simplifies the problem from, an unsolvable problem in genetics to a solvable problem in metabolism, because it reduces the number of targets, it reduces the number of approaches that you need to take, and it simplifies it to flicking a single switch, which is to turn off the ATP production. And the key is to do that without the toxicity. And with a drug like bedaquiline or doxycycline, you can do that because, you know, other people have taken the time to do the phase 1 clinical trials to make sure that they're safe and efficacious for their particular indication. All we're doing is taking that same drug and then repurposing it. But once you know what is the answer, then you can work backwards and you can find many different drugs probably that will target the mitochondrial ATP production.
Is the drug that you used, is it a generic drug or the Saturo, or is it still on?
I think it's still on patent, but it's only for drug-resistant TB, so it's not widely used. But doxycycline has been off patent for many years. It's been around 50 years. And we saw, it may not be as potent, but it did have the same effect in blocking metastasis, preventing metastasis. So I think the good news is that, you know, they're both mitochondrial inhibitors. They both ultimately reduce ATP production. And the idea is to kind of ATP depletion therapy. So you want to do what you can without toxic side effects to reduce the ATP levels to prevent the metastasis. It's a very simple idea, but it could be very powerful because, you know, if this turns into an FDA-approved therapy, then you would save, you know, if you could prevent metastasis, you would save potentially most of the patients that have cancer.
Wow, that's quite powerful. Now, does this kill off the ATP high line of cells? I mean, would this permanently prevent a metastasis? Because as you mentioned, it doesn't kill the tumor, so you'd still need some therapy to address, you know, the presence of the tumor in certain cases.
You certainly need to do more experiments, but as a first step, we see that these cells don't metastasize anymore. So we don't know if they're dormant. We don't know what happens if you take away the drug, although we have done experiments like that in the past and the whole cell line died. So I think the cell line ultimately depends on the cancer stem cells because they make new cancer cells that are not the cancer stem cells. They make also differentiated cancer cells. So you may need, and the other thing is that
so you're essentially putting a box around the tumor. You're saying, okay, tumor, you may be here, but you can't go anywhere else. We've prevented that part of the process of cancer, which, as you note, is the part that tends to lead to death. So that's rather important.
The other practical point is that normally most tumors are surgically excisable. So the surgeon goes in, takes out the tumor. The problem is when, you know, the surgeon has missed a few cells, and, you know, it's almost impossible not to miss a few cells. So it's those remaining cells that could metastasize. So you'd want to give the drug before or at the same time as you do the surgery, because then you could prevent the metastasis. So it's, and I
know there's been a lot of speculation that one of the primary causes of metastasis is surgery.
Exactly. So the inflammation, the wound healing, but most cancer drugs, you know, that were designed, they were designed to kill the tumor cells, but not the metastasis. So most cancer drugs actually inhibit tumor growth. But sadly, that's not the problem because the surgeon takes out the tumor. So the whole process of drug discovery has been hampered by focusing on the wrong stage of the disease. What we should have all been focusing on is on the metastasis, which is more mysterious. So it was easier to study tumor growth, more difficult to study metastasis. So now
the standard of care is mostly geared towards dealing with tumor growth rather than dealing with metastasis. Right?
Exactly. And the problem is that the tumors, that's not actually a problem, you know, because the tumor is removed. So the only problem is the disconnect between the approach. So all the drugs were designed to inhibit tumor growth, but there was no tumor because it was
you'd removed it. Right.
Exactly. And there was just the metastasis cells left, the metastasis initiating cells. So what we see, surprisingly, not surprisingly, is that, you know, what other people have seen is that anti-cancer drugs inhibit tumor growth but have no effect on metastasis. What we've seen is that bedaquiline and doxycycline have no effect on tumor growth but only inhibit metastasis. So we have the other half of the equation. That also suggests that we're right, because, and it also suggests that the process of metastasis is metabolically different than tumor growth. So it's sort of like starting over. That's the bad news. The good news is actually maybe we've figured it out now. I mean, obviously everybody will have to test this and validate it, but
So
it could be a window into how to prevent metastasis.
So back to the Warburg effect and reverse Warburg effect. The tumors are relying on the Warburg effect, and the metastatic cells are relying on the reverse Warburg effect.
That's what we think. And we actually had published before biomarkers for the reverse Warburg effect, and they predict recurrence and metastasis. So it all
And how does that work? These biomarkers.
For example, stromal markers that actually are associated with feeding the tumor cells, and mitochondrial markers in the tumor predict metastasis. So if you have more mitochondria,
I see.
the idea is that then those cells may have a better chance at leaving the tumor. So it all fits together. It's been a long time. I mean, we started working in metabolism in 2009. So it's taken 10 years for people to accept the reverse Warburg effect. And now we've sort of hit the nail on the head, the ultimate proof, which is the ATP and isolating the metastatic cancer cells. So I think this will be a turning point, because, you know, anytime in science when there's a new paradigm, it takes a long time for people to accept a new paradigm. And, you know, you need extraordinary proof to introduce a new paradigm. So I think
this could be a critical moment for shifting the perspective towards a metabolic approach to cancer, right? In terms of,
well, towards a more effective metabolic approach, let's put it that way.
Because before, people were really thinking about inhibiting glycolysis. Of course, that will also work because mitochondria are dependent on glycolysis for the intermediates that they need. So both approaches will work. But, you know, I think the thing that is different now is that before, even talking about inhibiting mitochondria was considered taboo. It was considered unfathomable, because the reaction I would get, you know, 10 years ago, or even 5 years ago, was that you're going to kill the patient. But that's not the case, because you can take FDA-approved drugs that inhibit mitochondria as a side effect. You know, people don't realize they're taking mitochondrial inhibitors like doxycycline. They know they're taking an antibiotic, but they don't realize they're taking a mitochondrial inhibitor. Just makes you feel a little tired.
Or metformin, which is one of the safest drugs out there and probably one of the most widely prescribed.
Also, metformin is a complex 1 inhibitor, mitochondrial inhibitor. So there's lots of drugs on the market that are already FDA approved that are mitochondrial inhibitors.
And are known to be safe.
So that people don't want to admit they are, but the data is all there in the literature. So of course it's okay to target mitochondria because it's safe. I mean, obviously we all have to deal with potential side effects. We have to find the window of therapeutic efficacy and the window of toxicity and avoid the toxicity, but it's not impossible to target mitochondria. In fact, it's been happening all the time. We just didn't realize that it was a benefit for a particular indication. Now we know that actually it's a benefit for preventing metastasis. So,
and even metformin's been proposed as an anti-cancer drug, if I'm correct in my recollection.
Right.
Although,
I don't know if they've done the trials with metastasis. They've done the trials with tumor growth, and they've had mixed results. But again, that would be consistent because, again, I think everybody was looking for an effect on the tumor.
Right, and metformin is inhibiting the mitochondria, not the glycolysis.
Right. So if it hasn't been done already, they should do the experiment with metastasis with metformin. And I think it would be beneficial, but it would be another potential drug. Obviously, it's not as safe as doxycycline, but because you can have lactic acidosis.
Right.
But I think doxycycline, the side effect of lactic acidosis is very, very rare, much rarer than metformin. So
I know we've only got, go ahead.
I was just going to say that I think the taboo is targeting mitochondria, but I think that's no longer a taboo because, all these experiments show that it is actually safe to do it with FDA-approved drugs that are already in use. So it's just a question of doing it how you do it. You're not going to use a hammer to fix a watch. You're going to use a very careful approach, like a watchmaker, to fix the watch.
I know you've got to go in a few minutes, but I wanted to ask you a big picture question, and in the last few minutes here, about what do you think in your research of cancer over these years? What do you think is the environmental factor that's driving this? If this is a metabolic issue, if there's a problem going on, why have we seen in the last century such a seemingly big spike and increase in these cases of cancer? That kind of gets at something that I talked to Tucker Goodrich about and Dr. Yu about, is what are these things driving this?
I think it's multifactorial. I think, obviously we're all living longer. And aging is the number one risk factor for cancer. So I think that plays into it. The other thing is environmental toxins. We're all exposed to more environmental toxins. When we think about plastics, for example, there's lots of things in the environment. All of these things can induce oxidative stress and can potentially mess up the mitochondria. So what we see in the reverse Warburg effect is the way that cancer cells fertilize the microenvironment is they secrete hydrogen peroxide. And then that's the signal for the fibroblasts to make food to feed the cancer. So oxidative stress is a key here, but we can't actually fix that with antioxidants because it turns out the ATP high cells have most of the antioxidants. So they use the ATP to fortify themselves with reduced glutathione. So they actually shield themselves from oxidative stress at the same time they induce it in their neighbors.
And doxycycline is an inducer of oxidative stress in cells, which may explain.
That's debated. It depends on, I guess, the dosage. But mitochondria, it actually acts as an inhibitor of mitochondrial protein translation. So you inhibit the 13 proteins that are made from the mitochondrial DNA. So then you essentially inhibit oxalates.
One of which is the tafazin enzyme, which the cell uses to recreate the electron transport chain when it gets damaged.
Right. And the other thing that we published that hasn't been extensively cited or taken advantage of is the fact that doxycycline is actually a radiosensitizer. So if you take doxycycline and you go out in the sun, you get a terrible sunburn.
Yes.
But actually, that's actually a great thing for cancer therapy because there aren't that many radiosensitizers. So you could take doxy, and we've done it in the lab. You take doxy before, give the cells doxy before the radiation, and it's 5 times, the radiation is 5 times more effective.
Right.
But it doesn't affect the regular cancer cells. It only kills the cancer stem cells. So we published that in 2015. That's something that should also be trialed. But you could imagine that would be another use for the doxy, the radiation sensitizer. So if the cancer cells ever became resistant to the doxy, you could still target them with the radiation. And we've already figured out the mechanism for that. There is a protein that confers radio resistance. It's called DNA PK. And it turns out that doxy reduces the DNA PK, I think, by 95% or something like that. So it's actually an FDA-approved a DNA PK inhibitor. It's not approved for that indication, but it is safe. So I'm looking forward to new clinical trials someday testing that in patients.
I see why David mentioned when you reached out to him about this new paper, how excited you were. I can see why you're so excited. This really sounds like it's quite a revolutionary step forward in trying to treat cancer, the most intractable of diseases.
You know, we're really excited, and I've already been invited to give a talk about it at MD Anderson. So that'll be in July. I won't be able to go there, but I'll do it on Zoom like we're talking now. But it's nice to have already some external validation on it.
Dr. Yu, I know you've been listening. Do you have anything you'd like to say or ask before we end?
Okay, just one simple question. So you mentioned about doxycycline, and this time you didn't mention azithromycin, so that's CPAC. So I know both are common use antibiotics, and in your previous paper you mentioned azithromycin have a lot of other effects, say anti-aging, or besides cancer, right? Anti-aging and even COVID. So why you choose doxycycline more than CPAC?
I guess doxycycline has been around a longer time. It's 50 years old, and I think also it's been shown that you can take it for longer periods of time. CPAC is 3 days and it's over. Doxy, you can take it for 6 months at a time with minimal side effects. So it's more safety also related to chronic exposure. So I'm sure they're both very safe drugs, but Doxy has a longer history of being taken for extended periods of time. So
which could be useful when you're dealing with something like cancer, right? Yeah.
Exactly, because you'd want to, if you were taking a drug to prevent metastasis, you'd probably want to take it for an extended period of time. Obviously, clinical trials need to be done, but that you need something that is relatively safe and efficacious at the same time, not something that's toxic.
Okay, so I see. It seems like got it. So doxycycline treat chronic disease because it can last for 6 months. But CPAC is only 6 pills for 5 days, so it has a surgical effect. Okay, just to let you know, my wife and my son both are caught COVID-19, and both tested positive and has a fever almost on the same day. And what happens is the second day, both of them take CPAC, and 1 day after, fever's gone. And after 6 days, 5 days dosage, the 6 days, all symptoms gone.
That's great. I'm not surprised, but it's great. I mean, a lot of people I know have benefited from azithromycin and doxycycline both. But that's great. But I have to run, unfortunately. Yeah. So we'll have to continue the conversation.
Yeah, to be continued. Thanks so much for your work. I really appreciate it.
Such a pleasure. Thank you so much.
Okay, thanks everybody. Great talking to you.
All right, we'll talk soon. Bye-bye.